Good morning. So Kaloko-Honokōhau National Historical Park is just located immediately north of us here, and it's an area of historical, cultural, and biological importance. The park covers over 1,000 acres of land and marine waters. And of 191 Anchialine pools that the park has surveyed, as many as 36, or about 20%, have been documented as having invasive fish species present. So this morning I'd like to summarize some of the field work we conducted earlier this year and trying to eradicate some of these non-native fishes from multiple Anchialine pools and pool complexes. So tilapia and these other invasive fishes that are found in the park pose a major threat to the Anchialine ecosystems via predation, competition, habitat disturbance. And one example, which Ron showed some slides of also, is the spawning pits that the Mozambique Tilapia excavate. That's one of the just more obvious types of changes. And then often we may just look at these other little fishes. You know, tilapia are moderate, medium-sized fishes. They're kind of stunted a lot of times when they're in abundance. But even these tiny ones are a threat to the Anchialine pool invertebrates. And they may appear tiny to us humans, but they are much larger than the Anchialine shrimp. And they're highly aggressive, and there's been papers that discuss how they either prey on them or completely change the 24-hour behavior of these shrimps. So actually, 2 of the 4 non-native fishes present in the park, the Mozambique Tilapia and the Western Mosquitofish, are on the IUCN's list of 100 of the world's worst invasive species. So the decision to use Rotenone in the field was a difficult one. It was a combination or a culmination of years of preparation. We did intensive reviews of the literature, consulted with many other experts and researchers. We did many field surveys in the park, and we conducted various types of lab experiments. And we also evaluated a wide range of fish removal options, various biological, physical, chemical methods, and combinations of methods. And we concluded that Rotenone gave us the highest likelihood of eradication success, could be achieved in days rather than years, and at a relatively low cost. It was also judged that the benefits of invasive fish removal far outweighed any minor short-term negative consequences posed by the Rotenone. Now, Rotenone is a well-known chemical, has been used in fish management for removal of undesirable fishes since the 1940s, so there's a massive amount of literature on the subject. It's a naturally occurring compound found in a variety of legume plants, as Ron pointed out. That's the active ingredient in auhuhu that kills fish. And all commercially available Rotenone formulations are composed of pulverized plant materials. So prior to the use of the Rotenone in the field, we conducted laboratory tests, bioassays, verifying that the Rotenone concentrations lethal to the non-native fishes would cause little or no harm to the native Anchialine pool invertebrates. And just as an example, here are some of the results from our earlier test, and this shows I want to point out the Opaula, which had 100% survival at all Rotenone concentrations tested, including concentrations above those normally used to kill fish. So now, Anchialine pools are amenable to the use of Rotenone due to several factors. These are really, a lot of them are very small habitats. The surface water volume is small, So the pool treatments require very small amounts of rotenone. In fact, Anchialine pools are substantially smaller than other water bodies where the chemical has been used. It's been used in sites, that are dozens, some hundreds of acres in size, successfully to eradicate invasive fishes. And at least here in the park, most of the Anchialine pools do not contain any native fish. Thus, the only fish facing harm are the non-natives. Now, there are definitely challenges that these small natural habitats pose. The hydrology of these pools are very complex. Water levels fluctuate with the tides. So the water volume of any one pool can vary considerably over a 24-hour period and over the course of the season or year. Each one is unique. Anchialine pools have rocky, irregular bottoms and shorelines, often with numerous cracks and crevices where fish can hide and take refuge. Anchialine pools have subsurface connections to other Anchialine pools. And there's this underground flow between the pools, which is also unique to the particular pools and highly variable, apparently. So to better understand the water dynamics, we mapped out the location of nearby waters in relation to any of the pools targeted for treatment. So here below is an example of one of those pools, Pool 128, represented by the red star. And it also shows the blue solid circles are some of the what I call the satellite pools. So there's about a dozen or so within 50 meters of the pool that we eventually treated. We also did some dye trace studies to understand the dispersal and the timing of the water, kind of as a surrogate for the rotenone, and also to verify that the rotenone would be retained in the pool long enough to kill the fish, because it needs at least a couple or 3 hours to stay in the pool to kill any fish present. So here are some of the basic results. So in total, we treated 16 different Anchialine pools, distributed them up among about 6 different clusters or assemblages of the pools. And of the 16, in 9 of the pools, just with a single treatment, we were able to eradicate all the invasive fish present. In 6 of the pools, 6 other pools, it took 2, in some cases 3 treatments to achieve eradication. And then only in the one pool, after multiple treatments, we've yet to eradicate those fish. Also, in most of the pools containing invasive fish, the Anchialine pool shrimp were absent or few in number in the surface water pool. Now, obviously, some of them, in many cases, they're living underground. In terms of shrimp recovery, following fish eradication, the auhuhu were commonly observed during daylight hours, often in high numbers. In some pools, it was noted that there was a dramatic change observed within days of fish eradication. The shrimp started appearing. However, in some other pools, we didn't see major increases in the auhuhu numbers until maybe over the course of several months or more. Let me run through a few case studies or examples of some of these pools that we treated. Here's a photo of Pool 123. This was before we did anything and before the non-native vegetation around the pool was cleared. Here's that same pool. The Park Service people cleared the vegetation away, so they're both to get rid of the invasive plants as well as to give better access to the pool. And then also the green then is showing the in February of 2022 this year the beginning of the dye treatment. And then about a week after we did the dye treatment, we did a rotenone application in pool 37. And we, all the folks on the right, we began netting and removing the dead and dying fish immediately. And here's my colleague Howard counting, measuring, and weighing the mosquito fish recovered from pool 37. And these fish shown here are just a small or a portion of the fish collected during that first hour. Then we would do follow-up samples periodically the first day of treatment and then over the following days. And here is a photo that the Park Service recently shot in early October of this year showing the abundance of native shrimp in pool 123. So this was approximately 8 months after the eradication of the mosquito fish. So moving on to pool 37, this pool contained both Mozambique Tilapia and Guppies. And the Rotenone treatment resulted in the collection of numerous Mozambique Tilapia and Guppies. However, within a day or two, the park people saw a couple tilapia swimming about somewhat happily, I guess. And the individuals that had apparently escaped the Rotenone by taking refuge in deep rock crevices. At least that was the likely conclusion. A few months after the initial treatment of pool 37, the park people in Megan conducted a second treatment and successfully removed all surviving fish and their offspring. And Kailua let me know that, prior to the invasion of pool 37 by tilapia, the pool had a healthy population of the Hawaiian native aquatic macrophyte Rupia maritima. The plant had disappeared when tilapia became established. However, in the months following our eradication of the tilapia, Rupia once again appeared in the pool 37. And apparently from propagules that had survived in the silty bottom of this pool. So that was cool results. Pool 54, which Ron mentioned, was the smallest, or one of the smallest of the pools in our study, but the treatment site, as Ron mentioned, was problematic because of rapid flush rate, and any dying Rotenone applied to the pool was found to be flushed out within just 1 or a couple hours of application. So consequently, our daytime application failed. Then we did a second treatment, and as Ron mentioned also, we also did use the commercial Rotenone in combination with auhuhu that we put in bags and stuffed deep into the cracks, and that proved successful. So you do have to adjust to every site how you want to handle these eradication efforts. You kind of learn on the job. Pool 18 is another one that Ron has already touched on. That was the first treatment that we relied entirely on the auhuhu plant. And we had just enough auhuhu from all that work to use it. Auhuhu use is a major undertaking. Plants were painstakingly cultivated, harvested, and stored over a number of growing seasons by Ron Englund in the Greenwell Botanical Garden. The material was processed by numerous individuals, me, Howard, Ron, and then everybody else joined in. And then when we treated the pool, we did recover numerous dead and dying fish, both tilapia and guppies, but again, as Ron mentioned, survivors showed up over the course of a certain period. And the second and third treatment of the site with both auhuhu and commercial Rotenone also failed to kill all the non-native fishes present. So the lack of success at pool 18, we believe, is due to several reasons. The site contains some really deep crevices where the fish can take refuge and escape the effect of the Rotenone. You can see here Howard holding this PVC pipe that we constructed with a funnel, and we stuck it as far as we could into the crevice and poured Rotenone in. Later, the Park Service actually used a pump to try to pump Rotenone into the cracks. We also likely undercalculated the pool water volume and didn't use sufficient quantities in retrospect. And again, depending on when you decide to add the Rotenone, it's going to vary because the tide effects. We also suspect that maybe some of these crevices actually have underground passageways that are possibly large enough for fish to move in between the pool and the adjacent Imakapa fish pond. Now, we're not 100% certain. Obviously, when we put the dye in, it moves into Imakapa and stains that water fairly quickly. But, you know, that doesn't necessarily prove that the pathways are big enough for fish to also move through. So what's in the future? Well, the Park Service has continued to monitor the pools and document certain factors that reflect on recovery. And the Park Service, I believe, are pretty intent on treating and retreating pools that contain non-native fish. Now, the big challenge is Imakapa. So Imakapa, if some of you visitors haven't seen it, you really should visit the park and walk along the beach to the sand berm and take a look at it. Because according to some experts, this 30-acre wetland complex is the largest Hawaiian example of an Anchialine habitat. It has a large reproducing population of Mozambique Tilapia. These were first discovered in about 2008, 2009. And it is a source and will continue to be a source of invasion to nearby Anchialine pools until they are eradicated. And so this here in this map, you can see the red triangles are ponds near Imakapa that had these have invasive fish in them that almost certainly came from Imakapa probably during high tides. The dotted lines are potential for the future if these tilapia continue to persist in fish, the Imakapa without being exterminated. So I'd like to recognize all my co-authors. Here are a few photos. Sally is gone but not forgotten. She's been instrumental in pushing some of this work. And then there are many other individuals who helped, and these are some of the individuals either helped in the field or helped process the plants. But I mean, I talked to a number of people to get feedback. David Chai, I'm not sure if he's here. Troy gave feedback when we had questions, so it's all very much appreciated. And this project was largely funded by the Park Service and USGS. And I'll say, Luamau Ki'io Oka'aina Ikapono o Hawaii. From it's the words is written on the first Hawaiian flag, and it's in the song Hawaii 1978. That brother is recorded. So, mahalo, thank you. Yeah, does anybody have any questions?
So I was interested, you mentioned at one point in your presentation that the shrimp were observed in large numbers during the day. Has any work, have you ever gone out and looked at night to see what happens to the shrimp in pools?
Well, the one common Opa'ula shrimp from my knowledge of the literature, I never can pronounce his name right, Metciolik, reported long ago that this is one of the few native Anchialine shrimp that is, pretty regularly seen during the day. And so, but when there's invasive fish present, they're gone. Now, I believe, that they do come out then at night because they change, these fish, if they're present, they change the behavior of these shrimp. They become more nocturnal. But in some cases, there's either no or very few observations of these shrimp. And obviously more can be done to, just to us.
Yeah, it might be an interesting student project or something.
And some of it might have already been done, and Troy would probably know better than me about those instances. Any other questions?
Do you know if Rotenone has any effect on waterfowl?
Well, there's been a lot of research on Rotenone, and there's the American Fisheries Society has put out standard operating procedures. The situation with Rotenone is a very unstable compound, and the reason why a lot of peoples from around the world, in Africa, South America, have used these Rotenone-containing plants to kill and stun fish. And you can consume the fish. I've consumed fish killed by Rotenone. The Rotenone that accumulates in the mussels is extremely low levels, and then it's such an unstable chemical that once you ingest it, your digestive enzymes just break it down. So there's not even any or much that even will get into your bloodstream. And the reason it's so effective against fish is that it goes directly to the gills, and it works at an intracellular level at the mitochondria, affecting, oxygen uptake. So it can kill them pretty rapidly at sufficient concentrations. Yes?
I was actually thinking about that, but I guess you answered. This is perhaps why you don't have to worry about the other species of fish that may use the groundwater, like anguilla and so on, because it's very unstable, the content.
Well, yes, and it varies. The stability of Rotenone relates to its half-life is reduced when it's exposed to sunlight, if the water's warmer. So in people that have used like Rotenone in northern lakes where it's really cold, well, it could stay effective for days or weeks, where here in more tropical countries, you would expect it to have broken down at least below levels that are lethal to fish within a day or two, maybe 3. Every case varies. And then organic compounds break it down, microorganisms. There's a number of different factors that affect Rotenone. Yes.
If it breaks down so quickly and it's so effective, how come it's so hard to get permits?
That's a good question. It's part of it's the big fear of using Rotenone or any chemical in the wild. So the EPA, long ago, Rotenone was used in agriculture, and because of that, it was listed as a pesticide, which means the EPA has to evaluate it every 10 or 12 years or something like that to determine re-registration. And for some reason, 12 years ago, they limited its use strictly to freshwater fish management. But the input that the EPA was getting input from was all these Western biologists in the mainland U.S. who were familiar, with using those streams. They did not get any input from marine biologists who were angry, who used it for sampling reef heads. And here, we're hoping that with this new re-registration that hasn't yet been printed, that they will have extended its use for, at least for areas like Anchialine pools, where you won't have to go through so much red tape. And we think the feedback we got from EPA that it is okay that we have that approval, but I haven't seen the release of their new decision. Yes?
Did you target particular tidal circumstances when you were doing your treatment to maximize the efficiency, like low tide, incoming, or vice versa?
Yeah, what we decided was to do our calculation and do the application based on right around the peak to right after the peak, so that then when you put the rotenone in, then it might like seep into the cracks and be a sufficient concentration to kill any fish. Obviously, if you put it in when it's rising, it was immediately going to get diluted. So, yes.
How long does the thing stay in there?
The rotenone?
Yeah, how long does it?
Well, again, it's each individual pool. So if you have a high flush rate, it gets flushed out and diluted to wherever that water goes. Obviously, if it gets carried to the ocean, I mean, you know, the amount of rotenone that you use in a pool would be minimal in ocean waters. Yes?
You observed movement of dye or materials from one peripheral pool into the pond. Did you ever observe reciprocal movement of materials? Like it was, you introduced it once, saw it here, and then saw it again in the original location?
Not in any that I had observed. I know the Park Service is contracting with an expert who uses dye at Ozark Underground, and we brought the guy with us, and the park really liked what he was doing, and they've used like different colored dyes. He talked about you could use different colored dyes, or you could add it to different places and then see if they merge somewhere. So I haven't seen that. At least, you know, obviously when the tide is dropping, it'll go one direction. In pool 54, which is that really small one, has a really fast flow, and the park people may be able to say something better, as the tide's dropping, it's heading seaward, and then as the tide is rising, maybe it does do a reverse flow and might carry the dye the other direction landward, but yes.
In Ecuador, right, to eradicate?
Yeah, we conducted a study in Ecuador. It has a big crater lake, auhuhu, which is about the size of Kamehameha. It's a old volcanic crater lake. It's much deeper than Kamehameha, and we used rotenone to completely eradicate Nile tilapia there. So that was good.
So it was bigger than, or about comparable?
It's about the same size. I'd have to go back. You know, the configuration, it did have lots of sediment, like that soft, silty bottom, just like Kamehameha, so you have to deal with that. And we did some little manipulations to dry the rotenone down deep.
Is there flow in that one?
No.
Okay.
So I guess I have 1 minute left. I'll take what's back there, please.
There.
Thanks. Thank you. Thank you.